So I will thank the, first I will thank the organizer. I will thank Vera, in particular Vera, to invite me to do this talk. Also, I would like to thank the CRCT to invite me. And my, so I will briefly summarize my career. I was a computer scientist initially, and my thesis is not on biology, neither on bioinformatics, but rather in high-performance computers, so high-performance parallel computer, and about the compilation, I mean the translation of high programming language into binary for high-performance computer. And then in 2000, I switched to bioinformatics, which is an emerging field at the time in 2000, and then I still be in bioinformatics. And my career will evolve from my first project was on sequence analysis, gene annotation, and so with one of my PhD students, which is now in Toulouse, which is now the researchers in Toulouse, not here, but in Ray. And then the idea of this, the basic idea is to try, instead of inferring the gene sequence and the gene sequence from a genome based on statistics, we try to mimic the human expertise. And the main difficulty is to translate into computational method this main expertise, and we do it with a representation which is like a knowledge graph. Okay, and quite surprisingly, in a world context, of comparing the different software on sequence, we won. We are the first, and we are three, and the other lab, there are 60 people, and so the basic idea that I think it is that we apply a non-conventional method compared to the method, and this reveal and this unconventional method will reveal us to be a winning solutions compared to the other. Software, okay? And so then I will switch, but I'm not really interested on sequence analysis. I mean, I am more interested in biological systems and systems networks, so I will quickly switch to systems analysis, and until now, I will stay in this field of system analysis. And the basic idea of this under the strange term of controllability is how to control the same fate by acting on, let's say, a biological network, which is the interactome. So basically this field is one of the field of precision medicine. The origin of this field will concern the precision medicines. And what is the precision medicines? The objective of the precision medicines is to have medicine at molecular scale, which is an emerging approach, a recent emerging approach for disease treatments. And of course, it has been powered by genomics data revolutions. I mean, precision medicines, the use of precision medicines certainly the breakthrough in precision medicines will be only based on the thanks to the omics data. So, okay, and then of course it is an interpretive approach of research, and which is quite surprising that if you have a deep look on precision medicines, you have a switch on the scale switch. A scale switch. Why? Because the causes is intended to be, so the precision medicine try to find molecular causes of disease. So the causes are at the molecular level, while a disease is often described in terms of symptoms meaning at a phenotypic level, And so the question is, what is the causal relations between a molecular level and a phenotypic level? Of course, this corresponds to a classical interpretation from genotype to phenotypes, but precision machines here will renew these relations focused on the disease. And then we try to start this talk by trying to investigate what these relations could be between, in one side, the genotypes, and while in the other side, the phenotypes in terms of disease. And then you have, in one side, of course, the different kind of phenotypes in blue circles, and in the left side, of course, the different genes. And if you are interested in the history of the interactions between phenotypes and genotypes, the first interactions, the first kind of interactions was the Mendelian rule around 80s. And then the Mendelian rules is a one-to-one, is basically, if you look the text of Mendel, is a one-to-one mapping between, on one side, genes and phenotypes. And here the causality is clear. Why it is clear? Because one gene is responsible to one phenotype, and then one gene is a cause of one phenotype. And so you have a clear understanding of both the causal relations between the molecular level and, let's say, the phenotypic level. However, some year after, you have the notion of epistasis, stressing the interactions between genes. And so then the causal relations will be not so straightforward, and these causal relations will be also complexified by the, let's say, pleiotropy, where one gene may be involved in different phenotypes. So back to our first questions, what is the genetic cause of disease by considering all these complex interactions? So do you have any idea of this? Or how can you find a model to clearly explain these relations by considering all the complexity of these interactions? The answer is quite simple, is simply a total mess. So by complexifying the interactions, by discovering the complexity of the interactions, so we are now unable to find these relations between, simple relations between, in one side, the genes, and in the other side, the phenotypes. And so it is very hard to tackle with a causal explanation in precision medicine to explain disease. So what do? The solution is quite simple. We try to integrate not one genes, but several genes interconnected in a networks. And then these networks will become the new referential genotypic representations to explain phenotypes. And the interactions of one particular the perturbation on one particular network will lead to particular phenotypes, and the difference in the networks of the difference in the networks corresponds to a difference to a phenotypes. And therefore, by doing this, by associating to different networks the different phenotypes, what do? Finally, we retrieve the basic interactions which that underpins the causality from molecular level to phenotypic level, let's say the one-to-one mapping between one representations at molecular level, the biological networks, and the consequence are in terms of phenotypes. So we want to be back into this one-to-one mapping simply to have this. And this corresponds to a new emerging field, which is network medicines. And the basis of network machines is to try to use the result of network science to solve problems, to have a grounding breakthrough in medicines by solving problems in medicine. And then what are the main topics of the network science, of network medicines? It deals with networks for disease analysis with two possible orientations. One is just topological analysis by looking at the topology of the network, the structure of the networks, while the other one is the dynamical analysis, looking for the evolutions of the network. Okay, and there is one worldwide institute now that that has been started in 2080, which is a network medicine institute that's trying to gather around the world different lab and different university in order to work on network medicines. Okay. And so the object of the network medicines is of course the Interactome. And as a computer scientist, the Interactome is interesting for me because it's not based on structures. I mean, genetic interactions is purely based on on structures and on the regulation activity and stress regulation activity. It's similar for protein. It's similar for other kind of signaling interactions, which is only represents the propagation of signal in a cell. Here in the interaction, will we gather different interaction types, so we are not focused on the structural on the structure of the interactions, but rather on the functionality of the interactions. And when we deal on functionality, it starts to be interesting for computer scientists, and this motivates me to pursue in the study of the interactions. So basically, there is two kinds of representations. One of the interactions. One is considered a protein-protein interactions, stressing the biophysical interactions between protein, while the other one is a combination of regulatory and signaling networks that compose the interactions. Okay. So from the interactions, the very question is, how act the disease on interactome? How the disease can act on the interactions if you do assume that this network will be able to explain the evolution of disease. And this question is quite simple by focusing on the structure of the interactions, which is merely a graph, which is rather simple. So what is a graph? A graph is a node interconnected with a link. It's a very basic mathematical structures. And if we focus on this, how can we perturb a graph? Actually, we can perturb a graph with four basic elementary actions that are called the stain, I mean structural action on networks, consisting on adding or removing a node or a link. And actually, we can see that there is a real correspondence with this elementary perturbations on a graph and biological actions, in particular in mutations. If I do consider a nonsense mutation, I can interpret it as a node deletions. While if I consider a missense mutations, I can, in some cases, interpreting it as a link additions because there is a new potential interactions that has been created with other kind of proteins. So what I mean here, I can find a strong correspondence between these four simple operations and some biological process, and more specifically in mutations. By doing so, I do something which is rather fundamental. I will switch for the disease explanation and disease mechanisms. I'll switch from complication to complexity. And then meaning that I switch for a large number of descriptions of disease to a very simple description of the disease based on elementary perturbations, and based on the difficulty is how to interpret this combination of elementary perturbation in order to explain disease mechanisms. And then I switch to something which is not computable. I mean, not computable, that means the explanation is only stored in databases and in book, but you cannot process it, to something which is computable. I mean, something that you can predict, you can do some method in order to predict the evolution of the disease or to predict some drug target, for instance. Okay, this is a basic switch by considering that only 4 elementary topological actions are the root causes of the disease and also the root causes of the treatment. So this is my claim, my working hypothesis, that the study of the stain in the interaction is sufficient for understanding disease and mechanisms and finding treatment. And here, for instance, if you have one perturbation, one non-decision, you will be ill. And then if you had another kind of perturbations, this will cure you. This will cure you. This is the cartoon of my talk. I'm really sorry for real physicians to show these cartoons. And so the stains are the basic elements to control the cell fate. So here, this is another sketch. You do assume that you have a normal cells. One mutation will go, the cell will go to a cancer cells. And a second mutation will cure the cells, or the cell will go in apoptosis. But here, the major problem here that for a modeling standpoint, I'm not at the same level. I mean, my consequences are expressed in terms of phenotypes, in terms of at the physiological level, while my causes are expressed in terms of molecular alterations, molecular perturbation. So my first step will be to switch everything at molecular level, and which is not surprising for physicians or biologists. I will express the phenotypes in terms of biomarker signatures. And then the basic idea, the fate control, the control objective is to reach a particular biomarking signature. Okay? So this is the basic hypothesis of my model. So once this basic hypothesis are stated, so I will try to really model the dynamic, and the rest of the talk will be how can I do to have a real model of this dynamic mathematically, and how to do some prediction based on this model. So I will focus on the model of dynamics, and I will focus on a very simple model, which is a Boolean networks. So basically, a Boolean network is a discrete-based modeling system, dynamical systems, where the value of the variables is 0 or 1. So it's a very, very, very simple networks. However, for instance, and how do we conceive a Boolean networks? Again, it's very simple. For instance, you imagine that these two variables, genes, G1 and G2, must complex together to fix to the promoting regions in order to activate the gene G in orange. So from the different possible input here, where the red disk stands for 0, while the green disk stands for 1, I try to predict the possible output according to this biological process. So normally I do this with students, and this is my question. So do you have any idea of what is the output? If, for instance, if you have 0, 0, what is the output? Is it 0 or 1? 0, thank you. It's okay. 0, 1, 1, 0, 0 again. 0, and again 0, and 1, 1 is 1. Okay? So for people in the room or for people behind their screens that did not know, that never do modeling, you do your first Boolean modeling. So congratulations. Congratulations. So this is the first modeling, and this table, it seems to be obvious, seems to be really simple, but we switch from description of biological process to mathematics. I mean, if I have this table, I completely describe a Boolean function. Okay, and from this Boolean function, I simply express it in terms symbolically, in terms of the operator, and this at mathematically represents the logical and where the only condition if the result is 1 is to have the both variables to 1. This is the definition of a logical and, but the other is symbolic. In terms of modeling, the only important thing is to be able to draw this truth table. I mean, describing the evolution of the function from the inputs. So basically, what is a biological Boolean network? Is simply a set of equations where each equation is a formula, is a logical formula, and this logical formula expresses the evolutions of the variable states in Boolean here. So this is one example here. This thing is a whole, and this is a negation. So you have a and a negation, and all. And then from this dynamic, we try to define the evolution of these dynamics. So this is the evolution of dynamics. How do we do? We simply how do we do? We simply for each state, we simply try to update one variable exactly. For instance, here. For instance, we have updated x1, so we go to state 0, 0, 1 to 1, 1, 0. Here we go to 0, 0. Here we go to 0, 1, 0 by updating the second variable x2, and then we update the third variable x3. Of course, it's a dynamical system, so the dynamical system will reach to some stable states, to some equilibrium. And the interesting point here for a discrete Boolean network is we have all the possible evolutions. By doing these representations, we have all the possible behavior of our systems. And one particular, so usually, for instance, in more complex modeling, that ODE modeling, differential equation modeling, we can only compute one trajectory. Here, by Boolean modeling, we can compute all the possible behavior, let's say all the possible trajectories. And here, one trajectory will be, for instance, by starting by 0, 0, 1, I will be a path in these networks. Okay, will be a path in this network until reaching a stable state, which is stabilized. So I can evolve here. So here, my modeling question is to try to find the biomarkers representing a particular phenotypes. Here, it will be translated. The phenotypes will become the biomarkers signature, and it is quite usual in Boolean networks, and it is an axiom that stable states represent phenotypes. So the basis is to try to identify some Boolean signature. Some Boolean signature at stable state here. So here this represents. So what does it mean? That this network represents a switch between two phenotypes. So the function is described as the function behind this Boolean network. The function supported by this Boolean network is expressed as a switch between two phenotypes. One has the signature 0, 1, while the other one has the signature, have the signature 1, 0. Okay, so, and then on Boolean network, so trust me, so you can do a model quite easily because you can do a model from different sources, literature or data, or gene expression, if I focused on genes, for example. Gene expression data from different scale, from different sources. It is easy to understand and design. I would say, to be a bit provocative, that even biologists are able to do Boolean, to do Boolean networks. So, because simply why it is easy to understand and design, because you can, there is a theorem stating that you can describe any Boolean behavior with the help of only these three basic operators, meaning the or, the and the note, and when the black disk corresponds to 1, while the white disk corresponds to 0, this is a truth table of this operator, and only with this three operator you are able to describe any logical behavior, meaning that any formula can be transformed as a combination of on, or, and not. Okay? It is a fundamental framework, and we switch quite easy. If you do estimate that 0, 1 is not sufficient, and you want to be more complex, you say, oh, my regulatory expression is more interesting if I can capture it with more than level 1, 2, 3, 4. Mathematically, we can automatically translate it into a one Boolean network that having the same signature. We published an article last year concerning these transformations between let's say multi-level discrete networks to Boolean networks. So meaning that your multi-level descriptions is just an interface to be processed with Boolean networks, and it's not a new theoretical framework. Okay, and then the framework is reliable. So why? Because in the context of the questions that you can ask, because of course you have only two values, and so this limits the set of possible questions. But in this context, and the set of possible questions are often related to the control activity, of the information propagation signal. And according to further questions, such kind of model, because they are simple, are relatively predictable. I mean, there is many articles showing that the model are reliable. I mean, they correspond to the reality. They correspond to the experimental reality. And of course, you can do predictions, and there is some prediction related to biological to Boolean networks, that have been done in literature. Okay. And so here, to represent the stain, we will extend the Boolean network to control Boolean networks. So what do we do? It's simply to add some new variables that will control this network. So, and then we add variables in the descriptions. So how do we do? How do we do this? I want to represent a gene knockout, which extract knockout here, mathematically. What does it mean, this knockout? It means that extra, when it is knockout, the value of extra will still be 0, and it will never evolve. So I will take the initial Boolean networks, and then I have my initial equation, and I will add this element involving parameters U3, okay? And then I will control it by deliberately setting U3 to 0. And remember, in the end, when you have 1, 0, the answer is 0. So if I set U3 to 0, of course, extra tend to become to 0. So by doing this trick, this mathematical trick, I can express the stain by the control network control quite simply. Of course, I can do it for link. I can do it with other expression, meaning by setting phrasing to one variables or a knockout or code. And if I change the value, of course, for any change of the value of the control, here I will switch off x1, here I will switch off x2, and here I will switch off x3. The dynamics will change. You just focus on the evolution of the dynamics by focusing on the equilibria. You can see here I have 2 equilibria. Here, by switching off x1, I have 1 equilibria. Here, by switching off x2, I have 2 equilibria, but they change. The equilibria are changed compared to the initial network, and then here I will have 1 equilibria in there. So by changing the value of some variables, I am able to change the dynamics of the Boolean network. So I am able to change the stable states and to change the things. And so in computer science, it is usual to focus on the questions and to focus on the queries. And then I will ask to the model 2 possible queries. One corresponds to the possibility, meaning that there exists one stable state at least having the expecting biomarking signature, while the other Y is for all stable state, I will have the biomarking signature. And this is the basic operators from which I am able to define the query to be solved like this, okay? And now, if I define what I am looking for, so I know I must answer to the question, how do I find the solutions? I mean, how do I find the appropriate control in order to go such that the dynamical systems will go to the expected biomarking signature, meaning a biomarking signature can be the concept, the signature of the cancer, for instance, or the signature of healthy states, for instance. Okay. And this is not so easy. So imagine you have a network of 100 genes. It's a big model, but not so big compared to human genome with 30,000 genes. So it's a big model, but not so big compared to the human genome. And you want to find the target candidates to cure disease, for instance, such that the number of gene inhibitions is 10 genes at most. So you can control, you can trigger one specific gene by setting it to 0, or 2 genes, or 3 genes, or 4 genes, and so on until 10. Okay, and so what do you do? The simple method will be to first it will be the simulations. I mean, I will switch off 1 gene, then I will simulate the evolutions by the evolutions. I will look at the stability, and I will look in my stable state if I find my signature. Okay. If it does not work, I will change the genes that I need to switch off, and then I will do with a combination of 2 genes, 3 genes, until 10 genes. Okay. So my question is, how many simulations you need to do? 1,000? 1 million? More? According to you. Do you have any predictions of how many simulations you need to do to cover cover all the possible combination space of control space? No, you don't know. Actually, it's more than 19 billion of simulations. Okay, And so if I do 1 second per trigger, 1 second per trigger, I need to have more than 600,000 years to finish. So if you decide to start a PhD with this, you need to start soon, okay? Okay, so what does it mean? It simply means that the simulation is not a good way to answer to these questions. And so why? Because I try to explain the method. So imagine you are in an archery tournament, and imagine that an archer, which is a blind archer, tried to throw an arrow to a specific target, which is here. So if I analyze this with if my only resources were archers, so I need to multiply my archer, so I multiply my trio, and then I need to throw blindly the arrow, and the result will be so I know that I will win only by the clamor of the crowd stepping there. Okay, and this corresponds to the simulations principle, which is in hypothetical deductive reasoning, considering as deductions. I mean, from the causes, which correspond to the archer, I will try to find the effect. Okay, and I need to compute in simulation. I need to compute this by simulations to show whether if my arrow will arrive to the target. So, however, there is another way, which is abductions. I mean, abduction is a relation from effect to causes, and this relation from effect to causes means that from the targets, I try to identify more targets here, and then by computing the trajectory reversely, I am able to find which archer will win. So this is quite easy from this, but you imagine that from in Boolean network, where the effect are only states, are only specific state at equilibrium, finding the control triggering, which is able to trigger triggering these states automatically. It's a bit difficult. I don't detail this, but this corresponds to these principles that I explained with archery. So we did it. We have a solver. We have a method which is called decipher, meaning choose your destiny, and it is based on prime implicant computation. And we have a software. Okay, and this is one part of workflow. With at the beginning we have an automatic network synthesis from data and literature. We can build a Boolean networks, and then of course we have planned, because this is not finished, we have planned to integrate to analyze the exposome in order to find the roots, the possible origins of perturbations. And then we have, of course, three which try to find the control in order to reach here the cure state, in order to treat the disease. And we also plan to have an analysis in order to overcome resistance. So this is a full workflow. The actual steps of the workflow that I've already done is one, the workflow synthesis, and three, control analysis, which is about to find the control. The other corresponds to future projects. And so I will end this talk by studying the applications on medicines. I will apply it to cancer. Why? Because cancer can be considered as network disease. Why I say that? Because simply the explanations of the application of the drug correspond or found is explained as the modifications of the signaling pathway as a perturbation of a signaling pathway in order either to trigger cancer or to cure the cancer by selecting appropriate target in signaling pathway. And the reasoning in cancer is like that. So it's not too difficult to explain in the cancer pool my method, because it's quite usual for oncologists to think like that. Okay, so we try to do this, and so we do a model. We did a model of breast cancer. We did of breast cancer, and this model, and this corresponds to the model, and this model has two stable states. One corresponds to divisions, one corresponds to apoptosis, because in cancer, this model represents the congruency between the cancer and the treatment. Why? Because if you remove the apoptosis, you have only the divisions here, and then you have proliferations and tumor, okay, and metastases. While if you remove divisions and you trigger only apoptosis, the cell will go in apoptosis, and then you have specialized anti-cancerous therapy. Okay, so this is a natural model in Boolean to represent the cancer and the model. Okay, and then we have two biomarkers, which is BAX and CIX1. And if it is off and if it is on, this corresponds to divisions, while the opposite corresponds to apoptosis. Okay, so first we decide to find the origin of cancer and to find the drivers. And then what is the query? The query is expressed like this. The query will be the loss of apoptosis. What does it mean? That means in any stable state, you don't want to find the signature of apoptosis. So we program this and we said, try to solve it, try to find the control such that at any stable state, I don't want to find the signature of apoptosis. And therefore, I have this result, and which is quite surprising for us, we are really happy to find that. We find actually all the causes of cancer that we have put in our network. Of course, it's a proof of concept, so we already know what are the origin of cancer, but we refine, so the software automatically finds them, and moreover, it segregates the oncogenes to tumor suppressor. And this is quite surprising for us. We don't expect to do this. This certainly means that the original model is not so bad. It's not so bad, but it segregated the source. And we have also other kind of predictions when you have a pair of controls that must be applied. And we are unable to check whether this corresponds to a way to trigger cancer by a setting, because we are in literature, and we don't find this directly. So we try to interpret something indirectly by the phenomenon of resistance. For instance, here they remark that if a GFR is inhibited, you have some kind of if a GFR inhibitor works, if and only which GS3K is not expressed, for instance. So indirectly we say, oh, it's not so bad, but we are unable to interpret them as the origin of cancer. Okay? And then when we have cancer, so we decide to set the Boolean network with cancer. So we use BRCA1. We fix BRCA1 to 0, so which is the origin of cancer, breast cancer, BRCA1 cancer, by inhibiting BRCA1. And then we again, so our question is try to get the apoptosis. So by doing this, we lose the apoptosis. Try to retrieve the apoptosis, and the result was, and you have different results. This result is very interesting because it shows that the method works. It says, okay, you set BRCA1 to 0, please set BRCA1 to 1, which is not a real ground breakthrough in medicine, but anyway, that confirms us that the method is not so clumsy. Okay, but anyway, we have this kind of predictions for this, and I will focus on the first one, which is part 1. She said, if you want to recover, apoptosis, fix to part 1 to 0. And this is really interesting. Why this is so interesting in terms of method? Because we find the lethal partner of BCR1, so which is really interesting in cancerology because you are able to have dedicated anti-cancerous therapy that only triggers the apoptosis in cancerous cell. So this is why it is interesting. And usually in other kind of method, it's not so easy to have prediction or to have prediction on the discovery on lethal partners. But here it's deeply involved or deeply integrated in our method, so we do not have any problem to find it. So we refine the lethal partner of BCR1, which has been which corresponds to the olaparib drug action in BCR1. That's why I do not explain the synthetic lethality here. So, but it is very interesting. So this shows that our method can be very interesting to find, let's say, complex drug targets and not so easy drug targets. Okay. So to conclude, so the cell reprogramming or the cell control, which has been done by network state controls that correspond to network state control. Our first biological validation I have done. Several validations are seemingly promising, so we try to pursue on this, of course. And the perspective, of course, collaboration are welcome with biologists and in order to apply this model. There is an extension. There is currently a PhD thesis of Jeremy Pardo, which deals with control sequence. Because why? Because, for instance, some cancer in cancer du côlon, colon cancer. You have a Vogelstein sequence, so and then the cancer is triggered according to a given sequence. So and it is the same for treatment. You have a sequence of drug also. So we try to examine the control sequence. It's more focused on the theoretical aspect now. So we try to extend it to the application in biology. Okay, and we are currently analyzing the secondary resistance. Because why? Because okay, the control, it's a control, but it's a control which has been counteracted by another control. So there is a concurrency between two agents, one corresponding to the disease, while the other one corresponds to the physicians, and such kind of model is unable to represent such kind of concurrency, and so we decide to investigate how to model it, and we choose game theory in order to try to model this, and we start to this. Thank you. When the visions around you